So these are my disclosures. I'm going to talk about stuff that's what I think tailored really to UK practice, so it's important to understand that because this thing is going to be on the internet. Now, Tom's already talked to you about some immunotherapy trials. I'm going to talk to you about a few others. We've just shared stuff that we found interesting. So have a look at this, Checkmate 003. This is a phase 1 study, and some years ago, a year ago, we were shown this curve, which is a long-term 5-year kind of outcomes for people who are put on nivolumab, a single-agent immunotherapy drug with lung cancer. And what we see at 5 years is 16% of people were still alive. And if you look at people with high PD-L1, tiny numbers, but it was a substantially higher number, alive at 5 years. Extraordinary stuff. So what was presented at this meeting that really I think was a key take-home was the 5-year long-term survival for the phase 1 initial studies of pembrolizumab. So this is the Keynote 001 study. So this is one of these multi-cohort phase 1b studies with multiple arms, but what we're talking about is patients with lung cancer given pembrolizumab, and there were 2 groups, people who are treatment naive, 101 patients, and 450, just under 450 patients who'd already had systemic treatment. And at the time of the analysis, which was November last year, median follow-up of 60 months, of the 550 people that went into it, 100 were still alive. So 18% of these patients are still alive, and 60 of them, 11% of them, 1 in 10 people starting pembrolizumab, completed a full 2 years, 35 cycles of treatment. Now these are the survival curves. Now these are very interesting. 5 years, 23% of treatment-naive patients are alive. In the previously treated cohort, which does have a higher number of patients, it's 15.5%, so fewer. Conclusion 1, there seems to be some importance of the line of therapy in terms of your long-term survival opportunities if you have immunotherapy. Now, this is exactly the same data, but split by PD-L1 expression levels. Let's have a look at the PD-L1 high patients, TPS score greater than 50%. So these are the patients that we would conventionally, until Keynote 189 got approved, think of giving single-agent pembrolizumab. At 5 years, 30%, 29.6% of patients are still alive. If you look at previously treated patients, it's 25%. It's not that much different, but it's a little bit less. So maybe line is important, but maybe if you've got really high PD-L1 expression, you might get away with it, not as much detriment. And if you look at the PD-L1 low, which is 1 to 49%, 15.7 versus 12.6. Not much of a difference, but again, slightly lower for previously treated. But these are the combined curves. So really important. What about toxicity? Well, what they looked at, we know what the toxicity profile of these drugs are. We've been using them in buckets of patients over the last 2-3 years. Nothing additional, no new toxicity signals at 5 versus 3 years, all the standard IO tox stuff. So what were their conclusions? The 5-year overall survival in Keynote 001 was 23% in treatment naive and 15% in previously treated. This is historical data from the US saying that with chemotherapy, standard cytotoxic chemotherapy, which includes Avastin in the US, 5-year survival is about 5%. So we're going from 5% to this new era of 20%. In high PD-L1 expression patients, the number of patients alive is consistently higher. At least a quarter of patients are going to be alive at 5 years. They looked at the 60 patients who had more than 2 years of therapy. So I now see people in clinic who are very upset at stopping immunotherapy at 35 cycles because they're doing really well. Doc, why am I stopping? And I have a difficult conversation with them. But I'm going to reassure them that actually 3/4 of these patients are still alive at 5 years. Really important data. No new updated safety issues. So that's that. Another trial. Immunotherapy, the IMPAL 150 trial. This is a complex but important study. So this is a drug, this is looking at atezolizumab in advanced non-squamous non-small cell lung cancer. And there are 3 arms to this trial. 1,200 patients, massive trial, 350 in each arm. And they're comparing 3 different things. First, look at this here. This is known as arm C. This is chemotherapy, carbotaxol with Avastin, a very standard first-line North American, some parts of Europe, treatment for lung cancer. So that's a good control arm with maintenance bevacizumab. This is the main investigational arm they're interested in, which is that plus immunotherapy, atezolizumab. So you have 4 to 6 cycles of chemo plus bevacizumab plus atezolizumab, and then when you have your 4 to 6 cycles of chemo, the chemo drops off and you stay on maintenance IO and bevacizumab. They had a third arm, arm A, which is very important, which doesn't have the Avastin in. And that's very nice because it gives you some signal as to whether the bevacizumab, which is an expensive drug, is actually doing anything in all of this. But the statistics of this study are quite complicated, and so it's not always been easy to tease out what this arm is doing. Now, this is the original publication in the New England Journal where they compared arm B and arm C, not arm A. This was one of the primary statistical things of this trial. Is arm B better than arm C, yes or no? And the answer is clearly yes, because the median progression-free survival goes from 6.8 to 8.3 months, and overall survival, again, goes up from 14 to 19 months. So this was a positive trial published in a very eminent journal. And in the subgroup analysis of PFS, they saw this interesting difference. About 10+% of patients had liver mets, and they saw that patients with liver mets seemed to have a more profound progression-free survival benefit than those without. So the company was somewhat interested in that. So this paper that was presented at the meeting last week looked at those patients with liver mets. And what they've done is they've shown with and without liver mets. Here we're looking at progression-free survival, and you can see that PFS with liver mets in patients who just have chemo with Avastin is not very good, 5 months, and it's a bit better with the addition of immunotherapy. If you look at the patients without liver mets, the PFS is a bit better, from 5 to 7 months. Tells you that liver mets are not very good. We know that. Visceral mets are bad news, and liver mets are very bad news. But there is still a benefit with the addition of immunotherapy to the chemo regimen. They also showed a comparison of arm C with arm A. And what you see is the inferior arm here. If you remove the bevacizumab and swap it for atezolizumab, they just kind of overlap each other with or without liver mets, which to me implies that there is something with bevacizumab. There's some sort of story of synergy in arm B of this regimen. It shouldn't be ignored. Look at overall survival. In overall survival, you really do see the detriment that liver mets give you. So without liver mets, median survival is 17 months. With liver mets, I mean, it's substantially less, 9 months. But again, the quadruple treatment arm seems to have a benefit. And when you compare the 3 drug regimens, be it with IO or bevacizumab, you don't seem to see as much benefit. So again, suggesting there might be a bit of synergy there. So what are their conclusions? What they're saying is that liver mets is a poor prognostic factor, which we agree with. That basically you do get higher response rates, duration of response with the 4-drug regimen in patients with liver mets. It's reasonably well tolerated, but you know, Taxol is involved in this. It's a drug we're not used to giving very much of in the UK because of its toxicity profile, dropping the blood counts and you know, quite substantial alopecia. So this is an option. They're not really saying that liver mets equals having this treatment. Well, I suppose they're trying to say that, but I think oncologists and the discussant would say what we know is that liver mets are a poor prognostic feature, and chemo-IO is certainly an option for those patients and will benefit them. So that's my second immunotherapy thing. This is a poster that I just thought was quite interesting. So this is from MD Anderson in Texas, and they looked at, I think it's 1,300 patients, just over, who'd had serial biopsies before treatment, after treatment, from different sites, and they were just mapping out what happens to PD-L1 in a patient. Is it static? Does PD-L1 vary by which site you biopsy? And they showed some interesting things. So PD-L1 does vary by anatomical site. So brain and bone mets tend to have low levels of PD-L1, and liver and adrenal mets tend to have more, so that they clock that. They also noticed that over time in the same patient, PD-L1 levels can alter. So after immunotherapy, repeat biopsies tend to show lower levels of IO, PD-L1 expression, but you do get differences in PD-L1 expression over time, and that heterogeneity does impact on the predictive value of treatment if you look at outcome curves. So I think this is just interesting. It just, all the time we sit in our MDMs deciding what's going to be biopsied, and my heart sinks when people start talking about doing bone biopsies, because there's often a vertebral met that's really easy to biopsy, but you've always got to remember that it's really difficult to get good molecular data from bone biopsies. So we just need to be thinking a little bit about this as we go ahead managing patients. So what do I think all of this means for the UK? Well, we're very lucky. We're very lucky in the UK, because for all our moans and groans, we can pretty much give immunotherapy across most of the indications that have been that we know about. So we have access to first-line IO, chemo IO, across all PD-L1 levels in non-squamous non-small cell lung cancer, Keynote 189, and the Empower 150 regimens also on the CDF. The rumours are rife that squamous chemo IO is imminently going to get approved, but I've just heard that in rumours. I don't know if that's true. We can still give single-agent IO to high PD-L1, Keynote 24. We can give any of the three main checkpoint drugs as a single agent in subsequent lines. There are some funny nuances to that. Pembro, PD-L1 expression, atezo independent of expression, nivolumab according to histology. We also have durvalumab after chemoradiation. So we're very, very lucky. But really, I think of this as a philosophical game changer. Long-term survival from that data and the Checkmate study, we've been giving chemo for years. 2, 3, 4% of our patients are alive at 5 years. Now we're talking 20%. It's orders of magnitude different. So now when we sit in clinic, the treatment of lung cancer is not chemotherapy. The treatment of lung cancer is immunotherapy. When you see a patient in clinic, you have to sit there and work out, how am I going to get this patient immunotherapy? That is the key point. Now, you may mix it with chemo first line, you may end up doing it second line. Whatever you do, if you don't give that patient immunotherapy, you're never going to see that outcome. Chemo is not going to do it. So we really need to get on this. We need to make sure eligible patients get access to immunotherapy at an appropriate time point. Now, there are all sorts of nuances to this. According to your PD-L1 expression, you may or may not be able to access it. There are PS, there are all sorts of issues about this, but it's really key to say that immunotherapy is the treatment of choice at some point. You've got to remember drop-off. If you decide you're not going to do it first line, you may never do it at all. And so we really need to get on the ball. We need to sort out how to manage these patients, not at a centre of excellence, out in the community, in the UK in general. So that's IO. Let's talk about another thing. So MET exon 14 skipping mutations. 3 abstracts. This is really interesting. So one abstract deals with a drug called capmatinib, another deals with a drug called tepotinib, and another abstract deals with acquired resistance. So what is MET? MET is another cell surface receptor with kinase activity. MET mutations have been documented for ages. They're known to be driver mutations, but they're complicated. They're not straightforward. Sometimes you get point mutations like you get in EGFR. Sometimes you get gene amplification. Sometimes you get this thing called an exon 14 skipping mutation. And sometimes you get fusions like ALK. And I'm going to specifically talk about exon 14 skipping mutations. So what happens is exon 14 encodes for the juxtamembrane zone of MET. And a whole series of deletions or insertions or things can happen that basically mean that this gets spliced out. It gets skipped. And if that happens, it causes a change in the structure of this receptor that impairs its degradation and ubiquitination and removal. So you get excess MET signaling because you've just got excess MET as a result of this skipping mutation. And it's important because it's actionable. There are drugs that have been developed to test it. It's not that rare. 3% of our lung cancer patients, it's as common as ALK. Patients tend to be older. They can be smokers or never smokers. They often have sarcomatoid or pleomorphic histology. It's a mutually exclusive driver mutation, and there are a whole series of different variants, genomic variants of these skipping mutations. So it's quite a complex thing, but there are drugs that inhibit it. So crizotinib was originally, which we use in ALK and ROS1, was originally developed as a MET inhibitor, but now we've got these new drugs here. And today I'm going to talk about capmatinib and tepotinib. So geometry. This is a phase 2 trial. Patients with an exon 14 skipping mutation, advanced lung cancer, EGFR ALK negative, good PS, get capmatinib. There's no randomization here. Everyone's getting the drug, but in 2 cohorts. A cohort of treatment naive and a cohort that have been pre-treated with previous chemo. The overall response rate in patients who were pre-treated was 40%. The overall response rate in patients who were treatment naive was almost 68%, much higher. The progression-free survival in the pre-treated cohort was 5 months, treatment naive, 9.6 months. You know, first-line people seem to be benefiting from this quite a bit. The stuff gets into the brain and seems to treat brain mets very well. 13 of these patients had brain mets, 54% of them had an intracranial response, and they achieved disease control in almost all of those patients. If you look at the different genomic skip mutations that you get with MET14, this worked across all of them. It seemed to be reasonably well tolerated. I mean, all these things that come up are quite difficult to interpret until you actually start using a drug, in my experience. But the thing that I always look at is how many people stop treatment because of a side effect, and it's about 11%. And a lot of drugs we use are around 10 to 11%. Actually, afatinib has something like a 9 or 10% discontinuation rate. All of the immunotherapy drugs, it's about 10 to 15%. So it's probably manageable. You know, these things report any toxicity at any time point of the illness. You can drop the dose. You can make things more manageable. So they conclude this is very active, high response rates, very good. Another trial using another inhibitor called tepotinib, which seems to be very specific for exon 14 skip mutations of MET. And again, advanced lung cancer, they're getting the same drug, and it's just a phase 2 open label study. And what they see is good response rates, overall response rate, 50%. The only thing that they split by in this is some patients had the mutation detected by a tissue biopsy and some by liquid biopsy. And you can see, you know, it's active. Again, a slight signal that first-line patients have higher response rates than subsequent line, but not as pronounced as with the capmatinib trial. And again, safety, they've had some lipase, fatigue, vomiting. The treatment discontinuation rate is 4 out of 87, which is low-ish. So they're concluding that this is an active drug. And then the third abstract is looking at acquired resistance. So they basically looked at paired tumor biopsies in people before and after MET inhibitor therapy, and they found a very complex architecture of resistance mutations developing. So some of them were basically on target, so further mutations in MET or amplification of HGF or new mutations in bypass pathways like EGFR and KRAS. So hugely complex pattern of acquired resistance. And so the discussant really talked very much about this observation of a benefit that earlier this treatment was given by response rate. So what does this mean? So I think it means we've got a new actionable mutation. Right? So currently we test for EGFR, ALK, ROS1, PD-L1, because those are the reimbursed pathways of treatment. BRAF V600E is not reimbursed in the NHS, but we know that the treatments for that are licensed and approved. But often if you use NGS technology to test for molecular testing, you will pick that up. We do have a series of investigational mutations for which there are targeted treatments that are currently not reimbursed, and I think MET's definitely in there now. So my opinion is MET exon 14 skip mutations are definitely actionable. And what does this mean? It really means we need to pick these patients up. It's the first thing to say. So there's going to be a switch to NGS. Our molecular pathway testing in the UK is a bit funny, with all sorts of people doing different things. But the NGS technology allows us to test for many of these mutations up front. And with the new genomic hubs that are coming online, we should be able to do that. We need to have a discussion about whether we should be using RNA-based technologies to pick up fusions, because NGS doesn't pick them all up. And I really want to highlight that the SMP2 program will pick up an exon 14 skip mutation in MET. And the Matrix trial has an arm of crizotinib that these patients can be offered. So even today, we have a pathway to treat these people if you can detect them. So that's MET. Let's move on to EGFR. So this abstract is looking at resistance mechanisms. This is from Memorial Sloan Kettering in New York. Now, in the FLORA trial, which compared osimertinib to first-generation inhibitors, they have published some data last year looking at Guardant 360, a liquid biopsy thing, in patients who are progressing on osimertinib. So we have some data already about resistance patterns in people who have had osimertinib. The difference with this is they're looking at tissue, not blood samples. So they did NGS on tissue before and after therapy, and patients were either first-line patients, so in the US you will give first-line osimertinib as a standard of care, but they also included patients who got osimertinib who were originally on first-line, first generation, and then got T790M. Not many patients, 27 had first line, 35 patients after failure of first line. And then they're just plotting what they found on NGS, on tissue examination. And there's some really important messages here. So first line osimertinib, patients biopsied on progression, there's a significant number who are getting transformation. So this is a histological change of the cancer into a squamous cancer or a pleomorphic cancer or even small cell cancer. Now, you will not pick that up on a blood test. You will need to do a biopsy to determine phenotypic change. In addition, a whole bunch of off-target mutations that we think are bypass signaling pathways that may account for resistance to osimertinib, and many of these are things we've been talking about today, and many of them are actionable, albeit not licensed, but in trials, and there are on-target mutations that can occur. And again, there are inhibitors being developed for many of these. What about patients who'd already had first-line TKI, developed T790M, and then were then treated with osimertinib? A different pattern here, which is really interesting. But again, histological transformation accounts for a sizable proportion of what's going on in patients, including squamous and small cell. You need a tissue biopsy to determine whether that's happened in your patient. Again, a whole series of often on-target effects, including the C797X mutation. And the incidence here, 28% of patients had a point mutation at C797. And that's really important because the FLORA Guardant data using blood suggests a much lower level. So on a tissue biopsy, you're more likely to pick that up. Now, we don't have an actionable pathway for these patients yet, but there are trials and drugs that are being developed that Sanjay was just talking about. So what they concluded is tissue rather than plasma revealed a high rate of histological transformation and different resistance patterns depending on whether you had it first line or subsequent line. So what does that mean for us? I think tissue biopsy post-progression in a mutation-positive patient is really important. It's the only way you can exclude phenotypic change. And as we get NGS online in the UK and we start using it, we need to be able to pick up the resistance pathways and give access of patients to potential new trials depending on what mutation we pick up. So now I'm going to jump to a completely different cancer, mesothelioma. So this is a randomized phase 2 study. Standard first-line treatment for mesothelioma is a platinum pemetrexed doublet. There is some published data for the addition of bevacizumab, but that's not licensed or reimbursed. So normally patients will get chemotherapy with carboplatin pemetrexed or cisplatin pemetrexed for between 4 to 6 cycles. So the question is, what about maintenance pemetrexed? Because we give maintenance pemetrexed to non-squamous lung cancer. So a logical trial to do is to examine the benefit of maintenance treatment in mesothelioma. And that's what this small trial tried to achieve. Unlike the registration lung cancer maintenance trials, they were all placebo-controlled. I don't think this was. But basically, the curves for PFS and OS are flat as a pancake. There doesn't really appear to be a significant benefit to either. The study was closed prematurely. Maintenance pemetrexed was well tolerated. What does this mean? I don't give maintenance pemetrexed. I don't know why you would in the first place. So that's all I'm going to say. Thank you very much.
Yeah, that's an excellent overview. You've really